All right, hello everybody. All right, cool, that works. Start my timer. So I'll be going back to the kind of protein coding world again. It's a 12-minute talk, so I'll go relatively quickly. I think everybody here can agree that protein sequence kind of determines function, but especially in the context of cell-based assays, and I think there's a kind of, something that's a little less often discussed, but actually also the expression level of that protein matters a lot in terms of the kind of output of what's happening at the cellular level. I think it's not necessarily discussed because, well, sometimes it's not an experimental variable. So when we're talking about prime or base editing of endogenous coding sequences, you're running with the expression level, presumably, of the cell model that you're working with. Other times, it might make sense to max it out. For example, with VAMP-seq, where you're directly fusing a fluorescent protein to your protein of interest, it makes sense to max out the signal for the wild-type protein because you're normally looking for kind of variants that are destabilizing abundance, for example, in which case you want the full dynamic range kind of going downwards. But if you're like me and you started playing around a lot more with kind of functional assays in cells, you end up realizing it might be a potentially important variable, especially in these kind of transgenic systems, for many of the reasons discussed are listed on the right, which I'm not going to get into today. This is actually super noticeable when you're using kind of like landing pad-based approaches. And I'll give a brief intro on that. So the basic kind of principle here is you've engineered cells with a single recombinase site. In our case, we use BXB1, kind of shown by that @P site and the star on that diagram over there. And also in our case, we then proceeded with a promoter, a 10^2 promoter for most of our constructs. And then in back of it is a series of cassettes that help you with all the cell engineering, whether it's fluorescent proteins, the actual recombinase integrates itself, or a negative selection cassette. And the idea here is, well, then you can, once you have these cells, you can transfect in a library of promoterless plasmids, each encoding a variant or of interest. Kind of complementary recombinase site in the plasmid, then using that integrase enzyme gets knocked into that site in the genome. And this only happens to one lucky plasmid. And the result is that essentially that one variant is now in back of that promoter, so you get single variant per cell expression. There's a bunch of benefits as well as some limitations to this. Again, not going to go into the details, but people on YouTube could, I guess, look at those. All right, well, so in starting to use the system and kind of exploring around the proteome, realize that, well, maybe we want control over the amount of expression that we're having, right? So just to kind of prove this point, or I guess demonstrate this point, in our kind of background 293 T cells that we've engineered, where there's no GFP, that's the background level of green fluorescence shown, measured by flow cytometry and shown on the x-axis. If we knock in GFP, we get very bright cells. They're also very homogeneously bright, shown as in this log normal distribution, which I think can be a boon in terms of kind of precision in terms of experiments. But what if we don't want that really high level of expression? In this case, well, I'll talk about that in a minute, but this high level of expression. One approach would be, well, we are using a 10^2 promoter. Why don't you start putting in kind of reduced levels of that inducer? And that works. The average level of kind of fluorescence within the cells of the population does decrease. But as you can tell, it's rather heterogeneous. Between that and the fact that we wanted something that was genetically encoded, we also wanted something that was genetically encoded in the plasmid library knocking in, so we don't have to rederive the cells. So for that reason, started playing around with essentially the Kozak sequence, which is kind of the informal name for the translational initiation sequence, which is the nucleotides, I guess, largely preceding the initiator methionine ATG codon, as well as some nucleotides in back. But this is the sequence that tells the ribosome to stop scanning and then start translating there. People have known of this sequence for a long time. So looking in literature, found some sequences that seemed like they would work less well. Tried those, and indeed we can reduce the amount of protein that's being made. But we're actually doing it in this kind of tight log normal distribution. So we like seeing that, and it made us wonder, well, can we use the Kozak sequence essentially as a rheostat to try to kind of program in whatever expression level we want? So to do this in a kind of comprehensive manner, we took a sort-seek approach where it was maybe one of the simplest libraries one can create, which is essentially 6 degenerate nucleotides in front of that starting ATG. Did a sort-seek protocol, which I won't go into the details of, but in short, just because we did this in front of a fluorescent protein, kind of sorted the cells with facts into 4 different bins, sequenced those, and created a weighted average, recreating the kind of effects for each of those Kozak sequences. So we get a distribution that looks like this, which is great, but this mean weighted average is a very arbitrary number, and I kind of wanted to get away from that. So what we did was we took 16 individual sequences spanning the range, expressed them individually in cells, and then essentially ran that in flow cytometer and used that to calibrate what our mean weighted averages scores actually mean in terms of flow cytometry terms. And that way, essentially, every single one of our sort-seek scores have been now converted into this calibrated, kind of flow cytometry compatible MFI value, which to me is a lot easier to kind of understand. All right, so how to say it look? Well, again, people have studied this sequence for a long time, so there's a lot of previous kind of information on it. People know that the -3 position is very important for the kind of efficiency of that translational initiation. That's indeed what we see in this plot I'm showing you with every kind of pairwise difference with A versus another nucleotide at each position. What is the kind of fold increase to translation that or steady state protein amounts that we're seeing? Yeah, indeed, the A is preferred there over C's and T's certainly, and slightly over G's. Great, that checks out. If you feed it into a kind of really rudimentary machine learning model, essentially a similar thing comes out with -3 being important, but also, upstream open reading frames are important. Again, something people have known about before, but it's nice to see it in this data, kind of validating that our data makes sense. Well, so the whole reason I even got into this was I was working on trying to do this tuning much earlier. And there's actually this nice paper from 2014, Noder et al., published in Molecular Systems Biology, where they actually do something very similar, albeit they do leave everything at these kind of arbitrary numbers, which I found very frustrating when trying to create my own experiments. But again, a very nice kind of comparable data set that we can compare our data set with. And we actually see a very nice correlation with a handful of exceptions. But those exceptions make sense. They're essentially ones with an upstream open reading frame where that -3 position for that upstream open reading frame differed between the two vectors. In our vector, it was an A. And then essentially the upstream open reading frame had a much bigger effect in suppressing kind of translation and steady state abundance of our protein that we were actually querying. And actually, we weren't trying to necessarily recreate what they were doing in that study. So there's a lot of different details here between cell backgrounds, the method it was put in. We took a much simpler sorting approach. But at the end of the day, again, I think the fact that it's so well correlative is suggesting that it's probably going to be a pretty generalizable phenomenon that could be used across kind of experimental kind of situations. So what are some things we can do with this information? Well, we can look at what's in ClinVar, for example. And we did this analysis like a year ago. We need to do it again. But we found essentially I think it's around 2,500 entries of Kozak sequence variants within ClinVar at that time. And then if we hone in on the ACMG kind of set that are particularly important, we definitely see some variants there. For an example, you can look at that first BRCA1 sequence up top where there is this upstream open reading frame, but based on our data, the suggestion is that it's actually reducing the amount of protein that's going to be there at steady state by 10-fold. So the idea is, well, maybe this person actually has, let's say, 55% of the BRCA1 protein that they might have made if they had two kind of reference Kozak sequences in front. All right, but we got into this more for kind of tuning or functional assays. So here are a couple of vignettes there. The first PhD student that joined my lab was an immunology student. She was interested in working on proteins and the kind of T-cell receptor activation transcriptional response pathway. So the protein she worked on is STIM1, and it's actually a pretty integral protein and kind of allowing the cell to be flooded with calcium ions, and this is part of the whole kind of biological mechanism for that transcriptional response. Because it's part of this kind of fundamental physiologic mechanism, there are gain-of-function mutations or variants in this protein that result in kind of aberrant hypercalcium influx, which causes tubular aggregate myopathy because it's important for muscle function as well, with this R304W variant being particularly toxic when we overexpress it in HEK cells. There are also loss-of-function variants where 2 copies of a variant like R429C result in, too little calcium influx and thus things like immunodeficiency because, yeah, T-cell receptor activation doesn't do anything. This is also kind of ER multimerizing protein, so I think there's some interesting kind of biology there, and you also see some very gnarly things, for example, with the R429C. If you tag it with M green lantern, overexpress it, you just see these gigantic paranuclear plaques. But anyway, in the case of Nisha, she wanted to do a kind of toxicity-based deep mutational scan type experiment. So if we started with just our kind of, our consensus Kozak GCC ACC ATG sequence in front of STIM1 and tried doing this experiment, we did see differential between the wild type loss of function and gain of function, but we just took this Kozak approach and kind of expressed these variants at this full 100-fold range of protein steady state amounts. And this, the data wasn't cleanest probably because of it being a toxicity assay through this particular biological mechanism, but we were still able to figure out that reducing the total protein that was being made by 30-fold kind of gave us the greatest kind of dynamic range in our assay between the gain of function and loss of function variants. We also have been doing a bunch of SARS-CoV-2, sarbecavirus viral entry type research. In that case, I'm not going to go into the details of it, but the idea is that we could modulate expression of the cell surface protein that is allowing the virus to bind to the cell and get in, where presumably the more protein there is, the more likely it is to get in, but that the relationship isn't exactly well known. Furthermore, incorporating sequence variants, we previously showed that that there are these kind of mutants that could be kind of engineered. Some of them actually, in the population of very, very, very low frequency. But this I21N variant increases or enhances infection. This D355N abrogates binding and essentially drastically decreases infection. In this case, yeah, so we essentially did this Kozak library approach in front of each of these variants, did it in such a way that we can test all the variants within a single experiment, and then challenge with pseudotyped viruses coded with different viral entry proteins. VSV is a negative control. It doesn't use ACE2 to get into the cells. We essentially see a flat line across all the ACE2 abundances, which makes sense. SARS-CoV and SARS-CoV-2 spikes, you do see that relationship as you would expect. That was a lot of data. I'm condensing it down into these overlaid lines. And I think it's nice and that it shows a couple of points. One, for example, a DMS performed at a reduced expression level, such as what I'm showing in that arrow over there, is going to allow you to kind of elicit some of these effects, like with that I21N infection enhancing variant that you wouldn't have seen if you were at that full 1.0 level. Furthermore, I think it's kind of a nice visualization of what I think can be in the future, which is, well, that each variant isn't necessarily a single data point, but instead it could be a dose-response curve, right? And that way, maybe we'll have a more kind of comprehensive, holistic systems biology view of what's actually happening in the cell. So yeah, in conclusion, I think that this approach is, or at least kind of modulating expression as with these assays, can make sense for kind of experiments in the future, especially when using kind of a landing pad type approach. And the next question is, well, should it be a separate preliminary experiment, or do you decide to put it as part of the main experiment, right? I think it's going to be very situation dependent. Also, do you want to test a full set of sequences, such as what we did over there, which was a library of 4,000 sequences? Or do you want to just test a few, in which case, here's a set of 6 that span that range where I've taken out the upstream open reading frames in either context because, again, that's going to be, very kind of construct specific. But I think the main point is, yeah, I think the Kozak sequence is perhaps a very convenient, very small kind of segment genetic tool that can be used to tune in whatever effects you're looking for in these overexpression assays, where if you're looking for a toxic effect, you might want to dial it to 100. Or if you're trying to do some, say a biosensor experiment where you need the cells to be alive to even indicate the experiment, well, it might make sense to tune it down lower to 10 or 1, and maybe that will get you in the right range. With that, I'd like to thank the people in my lab. I work closely with my wife's lab. We share adjacent lab spaces. I work with her, I work with her for all the virus-related experiments, although anything outside of virus, I kind of do on my own. But anyway, I'll stop there. Thank you very much.
All right. Very cool tool. Have a couple of questions, it looks like. I'm going to ask one first. Will this work sort of qualitatively and quantitatively similarly in different cell types?
Yeah, so I think the thought is yes. There are definitely, in kind of trying to prepare the manuscript, I definitely, went back through the literature. There's been work in a bunch of different, like kind of low throughput contexts in a bunch of other situations. I kind of tested our scores against theirs. I think it correlates pretty well. Even that Noter et al. One, it was in like a mouse pre-B cell or something versus our, again, heck, human cells. So I do think it's going to be a pretty fundamental thing that, again, should kind of work between cell types. Fritz.
Super cool. I'm excited about this. I want to try it. So I guess two questions. When you see an upstream ORF, are you sure that you're not tacking on a few amino acids, getting an early start on the protein? And then the second question is, have you tried TTG as a start codon if you really want to dial it down?
Yeah, good question. So I think I probably specifically looked at kind of out-of-frame start codons. I don't remember if we saw anything different from in-frame, in-frame start codon. There's not gonna be a lot in this 6 nucleotide segment. I can go back and look to see if there's anything interesting happening with the kind of 2 amino acids that were added on. But yeah, at least in terms of anything I showed, it was all out-of-frame stuff that was exhibiting that kind of decreased downstream translation effect. Yeah, in terms of kind of alternative start codons, definitely didn't look at that. I think that might be one way to kind of additionally modulate things. Yeah, there's a presumably 100-fold kind of range that can be tuned here. I suspect there might be strategies with which to kind of increase that range, whether it's kind of more explicitly incorporating dedicated upstream open reading frames or kind of incorporating additional start codons. We also didn't mess with the nucleotides after the start codon, mostly because they're protein coding. But I guess in a kind of more advanced setting, it might make sense to kind of modulate those as well.
Hey, great talk. So when we use the BXB1 landing pad in our lab, after a couple weeks after integration, culturing, we see silencing, or we believe to be epigenetic silencing, where the fluorescent marker we use starts to decrease. Do you think that with a lower expression level, with a different Kozak sequence, that it might deal with this issue?
I think my short answer is probably going to be no, with very little data to back it up. I mean, my interpretation is that there's essentially just like random, at least in terms of that effect. It's a common, we could do kind of landing pad troubleshooting discussions, both here but also later. Silencing is very common. My take on that is just like the genome is just randomly turning stuff on and off or whatever. And if there's no fitness advantage to keeping it on, then it'll turn it off. So essentially my advice for everybody is grow your cells, to incorporate essentially positive selection cassettes. So before even doing the landing pad kind of recombination experiments, culture your cells in BLAST, at least if you're using our constructs, because that will keep every landing pad locus active. And then afterwards, we usually have an IRES Puro cassette or something, and that keeps everything on. And that way, everything is essentially 95% greater pure throughout the entirety of the experiment.
Okay, great. And second question. What about potential alternative translation initiation sites in the gene? If you have a low expression sequence, is it possible that it would skip that and instead do a second translation initiation?
Yeah, I mean, I imagine that could very well be the case. Yeah, I mean, and I think even that kind of like differential upstream open reading frame kind of example comparing our data to Noter et al. Paper is a nice indicator of that. So yeah, I think there's going to be those relationships that are happening throughout that segment of DNA, and including some of the coding sequence, but if it's the coding sequence, obviously it's going to be very kind of experiment specific.
We might have time for one more, and then we should move on.
I got the mic, so I'm going to ask a question. So the idea of kind of building in a dose response to every scan is super cool. I love it. And I imagine you're playing around with kind of jointly modeling the dose response and the effect. Have you seen anything crazy. I mean, you sort of the assumption is you would just see shifting shifting sort of sensitivity to high abundance or low abundance variants. But are there any variants that behave paradoxically and keep you up at night?
Good question, and it's an easy answer, and I don't know, mostly because we haven't actually kind of expanded it out to larger experiments. But I think the idea is, yeah, we will kind of more incorporate this into many of our existing projects. Partially on necessity to, again, kind of try to get around some of these toxic phenotypes. At that point, I think we might have some nice examples that we might report back to you.
All right, well, let's thank our speaker for a great talk.